were two approaches that I kind of talked about in my presentation. One is I would say CAR T cell extrinsic approach, where we engineered microparticles that would help in the manufacturing process. So as we know, CAR T cells are taken out from patients, ex vivo, modified, and then infused back. And so this would be in that manufacturing part, where it would improve the way that these cells are manufactured to give a better product phenotype at the end of that manufacturing. And then and that would include enriching for memory phenotypes that have been shown to be associated with more durable responses in patients. And then the second part was a T cell intrinsic approach, where we engineered the CAR T cell to secrete its own antagonistic peptide against a novel immune checkpoint. And that approach kind of changes the way that the metabolism and activation profile of the cell, and so that helps us get a product which is more potent at the end of it. Before we can actually, make CAR T cells for patients that using this approach, we need to do some more GMP grade studies, so we can show that this approach can be used within a manufacturing facility to actually manufacture like CAR T cells for patients. For the VIP receptor antagonist CAR T cells, I think we'd have to we're doing some of the studies now to look at syngeneic models, so looking at mouse CAR T cells and an intact mouse. A mouse with an intact immune system and mouse tumors, to see these are secreting and they're found on other cells. And so two things: one, whether we can engage an endogenous immune response and we're getting that paracrine function, but also if there's any on-target off-tumor toxicities that might be happening from antagonizing this receptor. And so a second part would be doing toxicity studies with the secreting CAR T cells to make sure that they're safe for patients.